Assalamualaikum and hi, my name is Tasleema binti Shazahan from S5T1. I'm from KEDAH Matriculation College. Today I'm going to discuss about a subtopic in biology. Guys, what do you think about this picture? This picture is actually about an abnormal amount of hair growth over the body, scientifically known as hypertrichosis, which is Ambrose syndrome. I'm sure all of you know about this picture. This picture is about sickle cell anemia. And lastly, this picture shows that the girl is suffering from beta thalassaemia major, scientifically known as Cooley's anemia. From these pictures, can you identify what is the subtopic that we are going to discuss? It is Chapter 7, Mutation, 7.2, Gene Mutation. Guys, what is gene mutation actually means? Gene mutation is a change in the sequence of nucleotide bases in a single gene. It is also known as point mutation. It results in a change in the amino acid sequence and thus may change the protein produced. The different protein produced may not function as normal. The simple example of gene mutation is sickle cell anemia. Guys, do you know how many types of gene mutation? There are actually 4 types of gene mutation. First, base substitution. Second, base insertion. Third, base deletion. And fourth, base inversion. Base substitution is one or a few base pairs in the nucleotide sequences in genes is replaced with another base pair. It may cause changes in a base sequence and results in changes in codon. The second one is base insertion. It is the addition of one or a few base pairs in the nucleotide sequence in a gene. Third one is base deletion. It is the loss of one or a few base pairs in nucleotide sequences of a gene. Lastly, base inversion is 2 base pairs or more are inverted in the nucleotide sequence of a gene. For a better understanding, let's see this diagram. We can clearly see here that the base C is replaced with base T. The second one is the base A and A is inserted into the nucleotide sequence. And lastly, the base G, C and A is removed or lost from the nucleotide sequence. And base inversion, we can clearly see here that these base pairs are inverted into the nucleotide sequence. Okay, now are you ready to explore more about base substitution in gene mutation? Let's go. Effect of base substitution. Base substitution, as we all know, it causes changes in codon. Okay, first it is missense mutation, second silent mutation, and third one is nonsense mutation. What is missense mutation? Missense mutation It leads to amino acid changes. The altered codon codes for a different type of amino acid. It leads to a non-functional protein. The next one is silent mutation. This type of amino acid does not change. Type of amino acid does not change. The altered codon codes for the same type of amino acid. That's why it is called silent. No change in protein produced. Lastly is nonsense mutation. Nonsense mutation changes a codon to stop codon UAG or UAA or UGE. Codon is altered to a stop codon. It leads to a non-functional protein. For better understanding, let's see this diagram. We can clearly see here for silent mutation, the base A is replaced with base C, but the amino acid that codes for this base, this codon, is still the same, which is alanine for normal polypeptide chain, and for silent mutation also alanine. But for missense mutation, if the G here is replaced with A, it totally codes for a different amino acid. While for nonsense mutation, the C here is replaced with U. It codes for the stop codon. Okay, do you know how sickle cell anemia occurs? Sickle cell anemia is caused by gene substitution. Amino acid valine replaces glutamic acid at a single position in the protein beta strand. It causes missense mutation. This will cause abnormal hemoglobin sickle S shape. Hemoglobin S shape is stiff and tend to accumulate in the capillaries. This type of hemoglobin is not efficient in transporting oxygen. This hemoglobin will cause patient to suffer from anemia. Okay, comparison between sickle cell and normal red blood cell. From this structure of hemoglobin, normal red blood cell and sickle cell red blood cell, we can clearly see that this structure of red blood cell will flow easily into the narrow blood vessel, but sickle red blood cell will accumulate. This causes the other red blood cell cannot flow through the narrow blood vessel. Thus, the cells will not get sufficient oxygen to do their daily activities. This picture is an overview of how normal red blood cell and sickle cell anemia will look like. This picture shows how valine replaces glutamic acid in hemoglobin sickle shape. There are few symptoms of sickle cell anemia. Firstly, insufficient supply of oxygen to the organ. From this diagram, we can clearly see that the normal amount of red blood cell in normal person is much more higher compared to anemia amount of red blood cell in anemia person. This type of person does not get enough supply of oxygen to the cells. Physical weakness and fatigue. If the cells does not get enough oxygen, the person will get tired easily. Lastly, organ damage. If the sickle cell blocks the blood vessels, thus oxygenated blood cannot flow through this vessel to the cells. Thus, the cells will damage and eventually will cause the organ to damage. Now, let me tell you the overview of gene or point mutation. Gene or point mutation causes a change in nucleotide base sequence. This causes a change in codon, which causes a change in the amino acid sequence. Thus, a change in the protein or polypeptide produced, or abnormal protein is produced. Guys, it's time for a quiz. Who is ready? First question, a mutation that does not alter the protein production is called a deletion mutation, B inversion mutation, C silent mutation, D transverse mutation. I will give you 3 seconds to answer this question. Yes, you are right. The answer is C. Second question. The original nucleotide sequence is ATC CAT. The mutation one is ATC GCAT. What mutation occurred? A. Deletion, B. Insertion, C. Silent, or D. Transverse. I will give you again 3 seconds to answer this question. Yes, you are right. The answer is B. Okay, next we are going to discuss about frameshift mutation. What is frameshift mutation? Frameshift mutation is a mutation occurring when nucleotides are inserted or deleted from a gene and the number inserted or deleted is not in a multiple of 3. This results in the improper grouping of the subsequent nucleotide into codons. Many of these deletion or insertion start in the middle of a codon. Okay, guys, how does frameshift mutation occur? It occurs from the point of mutation. Nucleotides are improperly grouped into codons. This causes the gene to be read in the wrong 3 base groups codon. The reading frame is altered. Thus causes extensive missense. Extensive here means largely. The resulting polypeptide consists of an entirely new sequence of amino acids. Thus, a non-functional protein is produced. These are the examples of frameshift mutation. When the base A is inserted, it is totally codes for a different type of amino acid from the normal. When we remove the base G, it is also totally code for a different type of amino acid compared to the normal. Okay, what are the causes of base deletion? Base deletion causes thalassemia. Thalassemia is caused by the deletion of nucleotide base pairs for genes which control the production of the alpha and beta chains of hemoglobin. Major thalassemia causes serious anemia. There are actually 2 types of thalassemia. The first one is thalassemia major. The second one is thalassemia minor. Thalassemia major is scientifically known as Cooley's anemia. It is a severe form of beta thalassemia. It's because of the presence of two abnormal genes that causes either a severe decrease or complete lack of beta globin production. Thalassemia minor is the presence of one normal gene and one with a mutation. It causes mild to moderate mild anemia. This is quiz time. First question. I hope all of you can answer these questions. A frame-shift mutation ultimately ends up affecting the position of the A. breeding frame, B. nucleus, C. cytoplasm, and D. centrioles. I will give you 3 seconds. Yes, you are correct. The answer is A. Second question. What type of gene mutation results from the insertion or deletion of nucleotides and alters the entire amino acid sequence from that point forward? I will give you again 3 seconds. Yes, the answer is A. Congratulations. Thank you for watching my video. I hope all of you understand about subtopic 7.2 gene mutation.